Alright, let's go ahead and get started on our lab. If you go to your calendar, you will click on Lesson 1.05, Laboratory, and you'll see a series of things you need to read before actually doing the lab. And this right here is the lab report that you will be submitting via Dropbox, which I will explain later. So let's go ahead and start with reading the lesson introduction and learning parts about the microscope and how to handle it and how to use the microscope. So click on this first one. Go through that very first lesson, and at the end of that very first lesson you'll see student guide. Click on that. That student guide will tell you not only the learning objectives, but it'll also tell you how to conduct the lab itself. As you can see right here, it tells you to open up the virtual lab and so on and so forth. But we're not there yet. So we're going to go back to our lesson and keep on reading about parts of the microscope, how to use it, and so on. So just go through this lesson and read it all. Once you finish reading about the lesson, you will come across investigation using a microscope. And this little box will appear. You'll just click on that. And on page 2, you will see a link for the lab. Click on that link. That link should take you to this tutorial. Now I'm going to go over the tutorial rather quickly because during my class I'm going to show you how to do the lab a little bit better and a little bit slower. So click on view the tutorial. So grab the lens paper and click and hold the lens and then let go of your mouse and put it somewhere where the red circle doesn't appear. So see how it's pointing the finger? That's when you know you can turn it on. So once you see the finger, you click and you hold your mouse and drag it all the way to the right. So we're going to grab this nose piece and flip it off. Make sure it says 4X. We grab the slide, put it on the stage. So we're going to click, and then once you click with your mouse, you're going to move your mouse up or down to focus. Almost got it. Okay, there's the red. Now, how do I find the blue thread? Well, see this stage right here? So you click and you move it around. Oh, I kind of see blue right there, so I'm going to leave it there. And then I just need to focus it. Click and scroll up. Move the mouse up, or down. Oh, that was pretty focused. Oh. Again, grab the stage, move it around until I see the yellow. And click and move the mouse up or down. Okay, I can see it, it's right there. I'm not sure where this E is yet, but we'll find it. so it's kind of focused. So first I have to find this E by moving the stager around. I can kind of see, so I switched it over to 10x, if you can see right there. I'm just gonna move the stager a little bit. Find focus. Move the mouse up. Oh, that looks pretty clear. So now we need to go to high power, and it's on 40x, and move the stage around until we can find it. Oh, right there. I like to get a little bit of the edge right here, so I know how focused it is. A little bit harder. So what do we do next? Well, you have to open up your student guide, and we already did the tutorial. So viewing two prepared slides, remove the microscope cover, we did that, pick up the lens paper, we did most of this, and I'm going to scroll down to where we are now. Part B, view onion root tip. Place the onion root tip prepared slide under the microscope, and we have to adjust it. Make sure you start off on the lower objective piece, nose piece, in order to get it focused, or else it'll be too hard to focus and see it. If you go over to your lab report, on number 4 it says to sketch what you observe when examining onion root tip using low power lens and then the high power lens. Label the sketches. So you can print this and draw it and then scan it and upload it, or you can just draw it on here, or you can just write and describe what you saw. I preferably would like a drawing, but if that's not possible, it's fine. You can write it out. This is what we see under low power objective lens. And this is what a picture that you see in a high power objective lens. See how I have it under 40x? So now let's go to part C, viewing diatoms. Now again, you have to start on the low power, focus, and then you can go to the high power and focus. Be careful not to go too much scroll too much down into the actual slide, or else you can break the slide or damage the lens. And you're going to draw what you saw under the low and high power objective lens. Or again, if you can't draw it, you can write it. That'll be fine. All different kinds of diatoms, so you can draw whichever one you want. Okay, so then you will draw that on part 5. Sketch what you observe in the brine shrimp. And for number 5, when they talk about brine shrimp, they're just talking about the diatoms. So draw what you see under low and then high objective lens for the different types of diatoms. And then for hay fusion, we will be looking at little pieces of grass and other plant materials. So we will now prepare a wet slide using hay fusion. We have to use a pipette and place a drop of hay fusion mixture in the center. And then with the forceps, a lot of people miss this part, we will pick up a cover slip and place it over that drop of water that we put on this. There it is, just little spots. That's pretty much what you will be seeing. And this is what it would look like under the high objective lens. And that concludes our lab. Okay. Make sure you do all your drawings or write down all your observations. And then when you're done, I would prefer if you just highlight everything and you copy it and paste it into the Dropbox. Okay, so to submit into the Dropbox, you're going to go into Tools, Dropbox. And remember, this is Lab 1.05. And you click here and you paste it. And then you click Submit. And then I'll go in and grade it. And just to double check everything's in there, it's all good and in there. And you can also submit pictures if you want to submit some pictures that you drew. I prefer it to be copy and paste, but if it's absolutely not possible, then that's okay. Submit and thank you.